Good morning, everybody, and thank you for being here. I'm very excited to be here. We had a very exciting year in mesothelioma, 2016, really at ASCO. There were multiple presentations. So what I'm going to do is I'm going to give an overview of the agents that we have and that are available. And then we will have very exciting detailed lectures on immunotherapy and on dendritic cells. So let's see. Of course, mesothelioma is, of course, the cancer of the lining of the pleura and peritoneum. The diagnosis is made histologically. Sometimes it can be confused with adenocarcinoma. So we use the immunohistochemical markers that characterize mesothelioma. Since not a lot of pathologists see it because it's so rare. So immunohistochemical markers that are important are cytokeratin 5 and 6, calretinin, WT1. Those are positive. It's TTF negative and CEA negative. There have been several germline mutations that have been worked out in mesothelioma. We do know that there is a clustering of cases that is observed within families, and therefore there could be a genetic component. The important mutation that has been becoming more and more prominent is called BAP1. It's BRCA associated protein. It is a tumor suppressor gene that is missing in germline mutations. This could also be a somatic mutations that can be acquired. And in this particular individuals, the asbestos exposure is particularly damaging, and they're the ones that can develop the disease. So chemotherapy, this was sort of our first line of defense. And prior to 2003, all sorts of agents have been tried, and there have been no standardized therapy. 2003 was a big year for mesothelioma. There were 2 international trials that demonstrated that combination chemotherapy, cisplatin and pemetrexed versus cisplatin alone, were quite successful and very good. And both of these trials showed that there was significant response rate and an increase in overall survival. This was the trial that was done in the US. Pemetrexed and cisplatin versus cisplatin alone. It was a phase 3 trial where 456 patients were randomized into 2 groups. One group, 226 patients got combination therapy. The other group got cisplatin alone. And there was a difference in overall survival and progression-free survival. The combination arm had a 12 months survival, and cisplatin alone, a 9.3 months survival. So at that time, this established that the combination therapy was the gold standard. And this was a standard of care for many, many years, pretty much up until now. As you saw, the survival was 12 months with combination chemotherapy. So since 2003 up until now, the goal was to develop new agents to look at immunotherapy and to see what can be used in second line to prolong survival, and additionally, what can be added to the chemotherapy backbone. Is this working? So the first trial that was done, this was very exciting. This was done in 2015. This was a MAPS trial. So this was mesothelioma, Avastin, Pemetrexed, and cisplatin study. So this was a multi-center study that was very similar to the previous one, where Avastin vascular endothelial-derived growth factor was added to the chemotherapy backbone. There were 448 patients in the trial. Those were the patients that were candidates to receive Avastin or bevacizumab. They were not allowed to have any cardiovascular toxicity, and they had to be no more than 70 years of age. Those patients were randomized to receive Pemetrexed, Cisplatin, and bevacizumab versus Pemetrexed, Cisplatin, and placebo. They were treated for 6 courses, and if they had stable disease, they continued onto bevacizumab. And those were very exciting results that we haven't seen for many years. The progression-free survival, there was a statistically significant difference, 9.6 versus 7.5 months. And more significantly, there was a significant difference in overall survival, 18.8 versus 16 months. We have not seen those kinds of numbers for many years, so this was very exciting. And when we looked at all the subgroups benefited from Avastin, whether regardless of performance status, histology, gender, or age. And the quality of life was also not changed, and certainly was about similar, was not worsened with addition of the Avastin. So based on the MAPS regimen, this made it into the NCCN guidelines. And so for the patients that are bevacizumab eligible, the guidelines now recommend using the combination chemotherapy of cisplatin, pemetrexed with addition of bevacizumab, with following for 6 cycles, and bevacizumab can be used as maintenance therapy until disease progression. So several questions came out of the chemotherapy trials. The first one, can carboplatin be substituted for cisplatin? And the answer to that question is yes. This was answered initially in the extended access program, where patients were given those different regimens. And there was no difference in overall survival and total time to progression. 1-year survival rates were similar. This was recently confirmed by a very large by a meta-analysis that was presented at ASCO by the MD Anderson group. They looked at 3 large databases, Medline, MBase, and Cochrane. They looked at overall survival, progression-free survival, overall response rate. They had 5 studies. They had 11 prospective, 5 randomized, and 12 retrospective studies. And again, in 5 studies with comparative arms, there was no statistically significant difference in overall survival. The response rate was greater with cisplatin and pemetrexed, but disease control rate was the same. So the conclusion was that pemetrexed and carboplatin is not significantly different than pemetrexed and cisplatin. The next question that was also asked and is currently still not answered is whether pemetrexed maintenance is useful. And the study, again, is looking at trying to use maintenance like we use in lung cancer with pemetrexed after 4 cycles of combination therapy. And the study is to finish accrual in June of 2017. Moving on to second-line therapy, there is no currently approved FDA salvage therapy, and several options are available to us. None of them are excellent. There is Pemetrexed, where patients can be retreated at progression, gemcitabine and vinorelbine monotherapy, and of course that's why this is where we're looking at clinical trials. So multiple agents, multiple biologic agents, multiple immunotherapy agents have been tried, and I'm gonna, the agents that are highlighted in red, I'm gonna try and briefly review today. We will look at tyrosine kinase inhibitors and immunotherapy, and at miscellaneous agents like anti-mesothelin agents and ADI PEG-120. So the first study, the first agent that we're going to look at, the study is called LUM-MESO. The agent is called nintedanib. It is a triple angiokinase inhibitor, and it inhibits vascular endothelial growth factor, platelet-derived growth factor, and fibroblast-derived growth factor. Many studies were done previously, preclinical trials, and in vivo it significantly prolonged survival in mice and inhibited cell growth as a single agent and in combination with cisplatin. And it showed an additive, not a synergistic, but an additive effect when used together with cisplatin in preclinical trials. So this is the first abstract that I'm going to discuss from ASCO of 2016. It was presented by Dr. Scagliotti. They took chemo-naive patients from 25 countries. They had 87 patients in the phase 2 trial, and they're planning to recruit 300 to 450 patients in the phase 3 trial. So they're using the combination therapy backbone, cisplatin and pemetrexed, and they're adding nintedanib on days 2 through 21, again, every 21 days for 6 cycles. And if there is no progression after 6 cycles, patients will be given nintedanib, which is oral, or placebo. So currently the trial is ongoing. They're in the phase 3 trial, and their primary endpoint is progression-free survival. So this should be reported in the next several years. So we're going to switch gears, and we're going to talk about a different class of agents. We're going to talk about anti-mesothelin drugs. Most, as Dr. Cameron mentioned, most of these have been worked out at the NIH by Dr. Rafid Hassan. So several agents are currently available. So just to review this briefly, mesothelin is a cell surface glycoprotein. It is cleaved from a larger precursor protein, and mesothelin is located on the surface of mostly epithelial malignancies, even though it can be present on normal cells. So several mesothelin agents are currently in clinical trials, and we will talk about 4 of them. The first one is a monoclonal antibody against mesothelin. The second one is the immunotoxin, where the variable portion of the monoclonal antibody has been conjugated to Pseudomonas immunotoxin. The next one probably is very familiar to oncologists in the audience. It's an antibody-drug conjugate, which is very similar to TDM1. It's the antibody that is attached to DM4, or maytansinoid toxin. And this is the mesothelin vaccine. This is a vaccine against using the mesothelin antigen that has been using a Listeria vector. So the first trial that we'll talk about, again presented at ASCO in 2016, is called Artemis, after the Greek goddess of hunting. It's a chimeric monoclonal antibody that binds mesothelin. Dr. Hassan has done a previous trial. It's called emituximab with cisplatin and pemetrexed, and it had an adequate safety profile. The median overall survival was 14.8 versus 14.3 months without the antibody in historical controls. And what they were able to show that higher trough levels were associated with higher overall survival and progression-free survival in their previous trial. So the trial that they're doing right now, again, they're using different doses, and they're using the cisplatin and pemetrexed backbone, and they're adding emituximab at 5 milligrams per kilogram every 21 days for 6 cycles versus placebo. If there is stable disease, they're adding, the patients are going to go on to continue on emituximab weekly or until disease progression, and the other group will get placebo. The trial is currently ongoing, and we don't have the results of this trial yet, but this is something to watch for. This is again, the Pseudomonas toxin, which is a recombinant protein that links the antibody to the toxin. So initially, this was the idea that these are the mesothelin receptors on the mesothelioma cells. So the antibody would bring the toxin to the cells, and this was supposed to result in cell death. And even though originally they had very good results, they were limited by the development of the neutralizing antibody to the compound. So this limited their experiments. So what they were able to show, they were able to develop a regimen which is similar to a bone marrow induction transplant regimen of pentostatin and cyclophosphamide. And by priming patients with this regimen first, they were able to eliminate those neutralizing antibodies and go on with their trials. They were actually quite surprised the kinds of results they got from using this conditioning regimens. So they felt that not only were they eliminating the neutralizing antibody, they were also probably eliminating the T regulatory cells that were suppressing the immune system. And therefore, they actually were able to show, this is the water plot of their pilot trial where they had very good results. And this is the picture that Dr. Hassan has showed at a previous meeting where there was a patient with quite a bit of disease before treatment. It did not get much better right after treatment, but this was result at 8 months. And again, they're also, they have published their results of trying to add the immunotoxin to Pemetrexed and Cisplatin backbone. The next agent is the vaccine, the live attenuated Listeria monocytogenes vector vaccine. So the idea again is that it's a vaccine engineered to express tumor-associated antigen mesothelin, which is expressed in mesothelioma. It stimulates the development of specific T cells against mesothelin-expressing tumors. The phase 2 trial has been completed, and again, this was presented in ASCO. This was a frontline trial. The patients were given 2 boosters of the vaccines 2 weeks apart prior to getting chemotherapy. Subsequently, they got 6 courses of combination chemotherapy with cisplatin and pemetrexed, and then they got 2 more boosts of the vaccinations that were 3 weeks apart. And if they were clinically stable, they continued the vaccinations every 8 weeks. This was a small trial. They had 36 patients that they could evaluate. They had 94% disease control. They had 3% of complete responses. They had 53% of partial responses and 39% stable disease. So their median overall survival was 20 months. And their conclusion was that it was a well-tolerated vaccine. The plans were to go forward with the phase 3 trial, but I'm not quite sure what is happening with that because we were going to participate in that trial, and then suddenly it was not at UCLA anymore. So the final agent that I mentioned is the antibody-drug conjugate. Where again the antibody is attached to the DM4 moiety. And this is the trial that was presented by Dr. Rafid Hassan again at ASCO of 2006. This is called enitumab raftansen, 180 patients with biomarker staining for mesothelin. And those were the patients that progressed on first-line therapy, and they were given the antibody-drug conjugate every 3 weeks versus weekly navalbine, which is a week second-line therapy. And again, the primary endpoints of the study were progression-free survival, and secondary endpoint was overall survival. Switching gears a little bit, we're going to talk about another drug which is coming into trials. It is called ADP-PEG-20. So the premise and the concept of this drug is that arginine is an amino acid that is required for everyday life. Cells can acquire arginine by 2 pathways. There is an internal urea pathway that is internal to the cell and the cells use the enzyme called arginine synthetase. Cells can also acquire the enzyme externally. So some cancer cells lack the arginine synthetase, and therefore they rely only on the external supply of arginine. There is an enzyme which is called arginine deaminase that catalyzes the conversion of arginine into citrulline. So it depletes the supply of arginine. The native ADI that is present in the circulation is the half-life is very short and is quite immunogenic. A pegylated compound has been engineered, and that makes the enzyme less immunogenic and significantly prolongs its half-life. So ADI-PEG20 can be administered IM weekly and completely depletes the arginine supply. So arginine-deficient cancer cells are affected because they cannot get arginine anymore, and normal cells are not affected because they have that internal pathway. So this is an ongoing, it's a Polaris trial called the Atomic trial. It is an ongoing phase 2 trial where primary objective is overall survival, and it's going to hopefully progress into a phase 3 trial. So these are the treatment-naive patients that have been tested and are deficient for the arginine synthetase. Usually they have biphasic histology, and the AS1 deficiency is determined by the central lab. So they're randomized in first trial, and again they're given platinum Pemetrexed, and they're either given the experimental compound ADI-PEG20, or they're given the placebo. So UCLA is going to participate in this clinical trial, and it's due to open in the next several months. Now we're going to shift gears again, and we will talk about immunotherapy. This is very briefly, this is what happens on a cellular level. Tumor antigens are processed, and the dendritic cell presents the antigen to the T cell, and T cell, of course, is the main effector of the immune system. T cells recognize the antigen that is presented together with the mediated histocompatibility complex, and T cells generate an immune response. They CD28 antibody on the surface of the T cell binds to the B7 receptor on the dendritic cell, and here we see that the immune system is activated where tumor cells, the helper T cells, and cytotoxic T cells are produced and go out and generate the immune response. In order for the immune system not to go out of hand, there is a break on the immune system, and in 48 hours after the T cell activation, there is a protein on the T cell surface called CTLA4, and CTLA4 essentially competitively binds with CD28. It binds to B7, and it shuts down the immune system. So this is a normal pathway in normal cells to avoid autoimmunity, and it is a major negative signal. So the first agent that has been used in immunotherapy and has been developed is CTLA4 antibody, and this is the antibody that binds to CTLA4 and essentially does not allow the break that normally goes on to happen. And the blockade of this checkpoint provided the first evidence of improvement in overall survival and improvement in patients with metastatic melanoma, and this was done in 2008. Another pathway that is very important, and that is all where most of the immunotherapy trials and agents are, is the PD-1 pathway. And again, PD-1 stands for programmed cell death. There is another receptor on the T cell surface that is PD-1. This is a more specific receptor, and again, it's also used to shut down the immune system. So in normal circumstances, after the immune response has been going on and needs to be shut down, the PD-1 receptor is expressed, and it binds to PDL ligand that is present on the dendritic cell, and this acts to shut down the immune response. Tumors co-opt this pathway because they can express the PDL ligand on the tumor surface. And again, the presence of this ligand binds to PD-1 and shuts down the immune response that is generated by the T cells. So what does this PD-L1 expression mean? There are several studies that have looked at it. The first study was done by Mansfield at the Mayo Clinic, and the second study was presented at ASCO. It was done by an Australian group. So Mayo has done a retrospective study where they determined the expression of PD-L1 in 106 patients using the immunohistochemistry. And basically what both of these studies showed is that there is about a 40% expression of PD-L1. It is associated mostly with the sarcomatoid histology. There was only one patient that was desmoplastic. These patients have done worse. They were less likely to be offered surgery, which of course probably had something to do with their sarcomatoid histology. And this is the survival that they were able to show by the PD-L1 expression. Patients that were PD-L positive, it was only 5 months, and PD-L negative patients, it was 14.5 months. This study was presented in 2016. It used tissue microarrays and stained the arrays for PD-L1, CD4, and CD8. They had 329 patients. But basically, they showed the same results. They had 42% of the patients were PD-L positive, and PD-L1 correlated with non-epithelial histology, and they had worse prognosis. So again, multiple agents are in development trying to correct this problem. So this is ipilimumab and tremelimumab, CTL4 antibodies, the PD-1 antibodies, nivolumab, of course, and pembrolizumab have been approved for lung cancer, but these are the PD-1 antibodies. And these are the new PD-L1 antibodies that are currently being looked at, and avolumab has been FDA approved in urothelial cancer. So the first agent that was evaluated and that I showed you is the CTLA-4 antibody, tremelimumab. This was this was the first study that was done in 2012. It was the first author, and the person who developed these trials is Dr. Calabro from Italy. So his results were initially very exciting. He had 29 patients. He treated them from May of 2009 to January 2012. And what was exciting is that even though only 2 patients had durable partial responses, one response lasted 6 months, another one lasted 18 months, and he showed that he had 31% disease control in second-line setting with an overall survival of about 10.7 months. So there was a large phase 3 trial. We participated in that trial and actually recruited quite a few patients. So this trial took place from May of 2014 to December of 2014. And in that short period of time, 571 patients were recruited, which is very impressive for any trial, but especially for mesothelioma. And those patients, there were patients that could have had up to 2 lines of treatment. Primary endpoint was overall survival. They were treated with tremelimumab Q4 weeks for 7 doses, and then every 12 weeks versus placebo. And unfortunately, there was no statistical difference in overall survival, which was very disappointing, but that seemed to be not completely a new thing in this disease. And tremelimumab patients had IDUS type or autoimmune side effects or adverse events such as dyspnea, diarrhea, and colitis. And this was again presented at ASCO in 2016. So so the group, Dr. Calbero took this to a different level, and now they presented at ASCO again. They're going to open a NIBIT meso study where they're going to combine the CTLA-4 pathway with a PD-1 pathway. So again, they're going to enroll first to second line patients, pleural peritoneal mesothelioma, and they're going to give them tremelimumab at a much smaller dose together with an anti-PD-1 agent, durvalumab. And the primary objective is going to be immune-mediated objective response rate. Secondary objectives are going to be overall survival and disease control rate, as well as safety. Running through very quickly the immunotherapy trials. This was the first one. This was the KEYNOTE trial, PD-1 inhibitors in mesothelioma. KEYNOTE trials were the trials with pembrolizumab in different diseases. There was a mesothelioma cohort that had 84 patients. Those patients were PDL positive, and they received pembrolizumab 10 milligrams per kilo Q2 weeks for up to 2 years. Primary endpoint was safety, tolerability, and there was an overall response rate was about 24% with disease control rate of 76%. And importantly, there were no new safety signals with this regimen. This again was presented in ASCO of 2016, avolumab, part of the Javelin solid tumor trials. He had 53 patients that progressed after platinum and pemetrexed. Those patients were unselected for the PD-L1 status, and they received avolumab every 2 weeks until disease progression. And again, very similar overall response, stable disease control rate of 56% with the progression-free survival of 17 weeks. This is the compound that is called atezolizumab. It is anti-PD-L1 compound. So this is a compound that is against PD-L1 that is located on the tumor cell and on the dendritic cell. So it inhibits PD-L1. And this is a SWOG trial. And this group of trials is a little bit different. This is neoadjuvant patients. So these are patients that are potentially resectable. So resectable mesothelioma patients, chemo-naive. They get neoadjuvant treatment with cisplatin and pemetrexed. If there is no progression, they go on to surgery. And then they get maintenance therapy with atezolizumab for 1 year. So the plan is, if this particular trial, if the part 1 of this trial goes well, they're going to move on to the next phase of this trial, where their neoadjuvant treatment is going to be chemotherapy backbone plus atezolizumab. And again, if there is no progression, they're going to go on maintenance antibody therapy for 1 year. So, and what would be very interesting, they're going to have translational correlates. And this is an MD Anderson trial that is leading this in the SWOG group. I'm going to just introduce the idea of vaccines. This is the WTI vaccine that was studied at Memorial Sloan Kettering, SLS001. As I mentioned earlier, WT1 is a transcription factor that is expressed on mesothelioma cell surface, and immunohistochemistry is used in diagnosis. So at Memorial, they developed a multivalent vaccine using the WTI peptide sequences to stimulate the CD4 and CD8 T cell responses. And they had a trial. They presented this in ASCO in 2016. They had about 40 patients that were randomized. So patients were randomized to receive the adjuvant, the GM-CSF, and the SLS001 vaccine. And the other group received the adjuvant, the GM-CSF, and placebo. They did not receive the specific WT1 vaccine. So the patients, and I have to mention, they completed their multimodality therapy. So they completed surgery, they completed radiation, and they've already had chemo. So this was a good group of patients, and they were WT1 positive by immunohistochemistry. So again, these are the median overall survival was 21.4% in the vaccine group, and it was 16.6 in the non-specific vaccine group. The patients that had R0 resection had a 39.3 months overall survival versus 24.8 months, which again probably speaks more for surgical technique, as we've heard before, but maybe there is a signal here with the vaccine as well. So in conclusion, there are important changes in the management of mesothelioma in the last few years. The role of VEGF has been elucidated, and probably we found the correct backbone, since the previous trials with gemcitabine and cisplatin didn't work. Of course, we need to focus our research on biomarkers and the pathway. The role of immunotherapy is more and more promising, and I'm very much looking forward to hearing today about the vaccine therapies. Thank you.